Michael Farzan, sorry, let me get this going, is our invited speaker today, and he is the chair, a professor and chair in the Department of Immunology and Microbiology at the Scripps Research Institute in Florida. And before that, he actually had a very long and illustrious career at Harvard Medical School before moving to Florida. So Dr. Farzan works on several interesting topics: HIV pathogenesis and mechanisms of preventing infection, which is a topic that I have never worked in directly, but have been very interested in. And then he's also had a series of really, really nice papers on the original SARS, so SARS-CoV-1. And yeah, this, I'm sure that you never really thought that we would be living in a pandemic about 16 years later, huh? So Mike told us that today he's going to share two stories with us, HIV and SARS. So we're really, really looking forward to hearing that. And thank you so much for virtually visiting us.
Great. Thanks, Ian. My pleasure to be here. Now I'm going to go through this motion of trying to share the screen. And so we'll see how far I get. There we go. And let's see. And now I'm going to presume that you're seeing what I'm seeing and make it full screen. And I'm hoping that that you're seeing my sort of title slide, where what I'm doing is introducing, I'm guessing somebody else, yes, a thumbs up. Thank you, that was helpful. And what I'm going to be talking about, as Ian said, is two stories. Oops, excuse me, looks like it's on a timer. The first story is about our work on HIV, and the second story is about our work on SARS, originally SARS-CoV-1, now SARS-CoV-2. And both of them sort of have the structure. We start from sort of basic science and work our way kind of to technology and almost, and I sort of apologize for this, to product development. And so you can kind of, if you're more interested in the science, listen early. If you're more interested in the later steps, listen late. But there are two, hopefully short stories that both happen to have that structure. So let me start with the HIV story first, and to do so, I kind of have to present how HIV gets into cells, and I'm going to do it with a little bit of bias toward, and I hope everybody can see my pointer. Usually they can. Bias toward tyrosine sulfate, and in particular these, which are these things here, which are three post-translational modifications of CCR5. They also happen to be post-translational modifications of the other HIV co-receptors. So I'm going to kind of give you a view of HIV entry a little bit warped in the direction of what the sulfates are doing. So we know, and many people who study HIV have heard, that the first thing that really happens, the first real action that happens when HIV is near the cell is that the cellular receptor CD4 associates with the envelope glycoprotein. So CD4 in red here, talking to the envelope binding site again in red. And that binding opens up the envelope glycoprotein trimer, sort of changes the conformation of the envelope glycoprotein. And that in turn allows exposure of the co-receptor binding site. Now, the next thing that happens is that these sulfate tyrosines at the immunoterminus of CCR5 or CXCR4 or co-receptor of choice, say in primates, directly interacts with the envelope glycoprotein, and in fact, as I'll show you in a second, kind of wedges its way under the V3 loop of the envelope glycoprotein. Stabilization of that V3 loop allows it to then, excuse me, it's really jumping the gun on me here. Stabilization then allows for the V3 loop to poke into a cavity of CCR5. And then this association promotes a series of fusion events that I kind of gloss over because they're not so important to my story, but they do involve insertion of a hydrophobic fusion peptide of GP41, the subunit domain labeled in pink here, into the membrane. formation of 6-helix bundle is a series of coils that provide the energy that drives mixing of the viral and cellular lipids that don't otherwise want to mix, pore formation, and entry finally of the viral capsid into the cell. Okay, so that's kind of the full big picture of the thing. And then what I'm going to do is kind of zoom in on that moment when CD4 is bound, the HIV envelope glycoprotein is open, and CCR5 is engaged. And that is this here. So this is what I've done here is combine two really beautiful structures from Andrew Ward and from Bing Cheng. So Andrew did the CD4 bound open configuration of HIV envelope glycoprotein. And Bing did the GP120 component, just one of these sort of tan things here, in association with both CD4 and CCR5. And if you can combine those, you can kind of get a picture of this stage of the viral fusion process. And what you see again is CD4 binding. And then now CCR5 is in cyan. I changed colors on you. And then these green things are all the glycosylations, which I just removed. In order to show you the positions of the 3 sulfotyrazines that bind CCR5. And one of them sort of engages CD4, tyrosine 3 doesn't matter. Tyrosine 10 lives in a very conserved region of the envelope glycoprotein. And tyrosine 14 also lives in a conserved region at the base of the V3 loop. And you can see that V3 loop now poking and stabilized and poking into CCR5. And that's the necessary event for the subsequent viral fusions. So CCR5 is very important. And so this is kind of a long story short on what we've been working on. The thing we've been working on is called eCD4. And what we did was we took CD4, a well-known inhibitor of HIV, a well-known but not effective inhibitor of HIV called CD4IG, which is just the first 2 domains of CD4, first 2 immunoglobulin domains of CD4, fused to an antibody FC domain. And then at the base of that, we put something that also encoded a pattern of tyrosine sulfates that mimic the patterns of tyrosine sulfates observed with HIV co-receptors. So that's the red thing on the bottom. And now I'm just going to give you sort of a list, and I'm sorry about this, but just to make this quick and kind of get to some of the interesting points, I'm just going to tell you what's good about this thing. So it's broader than any neutralizing antibody, and that's important. The main problem with neutralizing antibodies is escape. And so this neutralizes 100% of every isolate, including SIV, all the SIVs we've tested, and HIV too. So it basically hits everything that so what we describe it as 100% breadth, whereas the best neutralizing antibodies might have so-called 95% breadth, and a number of neutralizing antibodies that some people in the HIV field may be familiar with have as low as 60% breadth. So of course that's not useful if you're going to be using that as a therapy, as we're learning from COVID cases as well. But it's, in addition to breadth, it's also harder to escape, somewhat dissociable concept from breadth itself, as we've been able to show in vitro and in vivo. It's as potent as most of the neutralizing antibodies. Maybe it's ADCC. It destabilizes the envelope glycoprotein, unlike neutralizing antibodies, because it's basically essentially fooling the virus into thinking it's in the process of fusing. Because it opens up the shape of the envelope glycoprotein that are unmasks novel epitopes, including epitopes that infected people with antibodies against too. We can make it very, very directly. And as I'll show you, it provides a very effective prophylaxis, at least in non-human primates. And it is potent enough that if we get the levels high enough, it can suppress and establish at least SHIV infection. And finally, that these proteins, analogous proteins, meaning ECD4 without the sulfopeptide, have been shown to be safe in both adults and humans in the past. But its major problem, and now we're kind of moving into the product development form of this, is that it has a shorter in vivo half-life than most antibodies. And I'll just sort of insert the comment that most of us academics rarely worry about the pharmacokinetic properties of the proteins we develop. But recently, and this is new in my career, have had to. And it turns out to be more complicated than just getting something that's very potent. So we undertook that effort, and we made literally hundreds of versions of this thing, and I won't belabor this except to say we optimize both the CD4 domain, primarily by stabilizing it. It turns out that CD4 is not a protein that is meant to be in circulating freely in your bloodstream, and it has a little bit of fluidity within the immunoglobulin domains. And if you stabilize those, they number one, bind HIV envelope glycoprotein with higher affinity, but number two, live longer in the bloodstream. And that's, I'm sorry, this jumps on me. And the other thing we did is we replaced our previous sulfapeptide, our peptide containing the tyrosine sulfates that mimic the co-receptors, with a series of sulfapeptides that were derived from other tyrosine sulfated human proteins. So they'd be less immunogenic and they happen to have better PK properties. And the result is we were able to, using a fairly established model for PK, which is this human FcRn mouse model. And the FcRn is a protein that allows antibodies to not be degraded in endosomes, but rather to be recycled out to your bloodstream. Using this model mouse, we were able to show that, that we significantly improved our ECD4 variants. These are now called these EMMs, so it's yellow, green, and blue, and getting close to the half-life properties and of our gold standard here, which is this very long-lived HIV neutralizing antibody called 101074, and surpassing more conventional, more average HIV neutralizing antibodies like VRC01 or 3BNC. All of these, by the way, for those who know this stuff, have an LS mutation. That LS mutation is something in the FC domain of ECD4 and these antibodies, that it enhances its affinity in one state for FcRn and significantly expands the lifespan of these molecules. So these molecules can last in humans up to about 2 months or so. The half-life is about 2 months. We also showed that we also significantly improved the potency of this, and that's just here. You can see the comparison with VRC01, which is a sort of a standard milestone HIV neutralizing antibody, and so it's generally a good thing. And so so now I've sort of skipped over a lot of the properties of ECD4, sort of listed them as sort of a spec sheet, but I wanted to linger on one of them, which is the ability of ECD4 to work in parallel with antibodies in infected individuals. So what happens in most infected individuals is they make antibodies against epitopes on HIV envelope glycoprotein, on GP120, that are normally occluded on the closed normal configuration of the HIV envelope glycoprotein. And what we're showing here is that ECD4 opens up, as you might expect, opens up the envelope glycoprotein to expose them, and thereby collaborate with actual pre-existing antibodies in patient serum to mediate antibody-mediated killing of infected cells. And so the way we're showing that is we're making antibody ADCC. ADCC is the activity that I'm describing, the cytotoxic activity, and we're making versions of that that are inactive. We're making versions of the protein ECD4 that is ADCC inactive, and then versions of HIV neutralizing antibodies that are similarly inactive. And what we're doing then is we're incubating them with patient sera, which has weak ADCC activity. And what you can see is that the ADCC inactive forms of the neutralizing antibodies don't change the ADCC activity of patient serum. So I'm showing you 6 examples where this is working down at the bottom here. And whereas ECD4 significantly, by a couple of orders of magnitude, enhances the ADCC activity of this patient serum. And we, in the paper where we described this, went on to show that this is simply for very understandable reasons. We're opening up the envelope glycoprotein in a way that now these other antibodies can come in and bind and help NK cells come and kill these cells. And so this is the only sort of caveat on this is there are patients, especially patients for, I'm showing you up top, this serum 9126521, which is an individual who's been on antiretrovirals for a long time, and they don't make these antibodies. They're no longer making these antibodies. This trick is no longer valid for them. So, if you wanted to get that same kind of effect on those individuals, you kind of had to vaccinate them with a bad vaccine. Not hard to make a bad vaccine. But anyway, so that's the kind of acute aspect of this antibody, which sort of makes it qualitatively different from other HIV neutralizing antibodies. Now, what I want to do now is go to the in vivo studies, non-human primate studies, that we've done with the rhesus form of this molecule. And what we typically do is we introduce this molecule through intramuscular injection with AAV, typically AAV1 or AAV8 capsids, and we combine that with an enzyme that adds the tyrosine sulfate at about a 4-to-1 ratio. So 4 ECD4 and 1 of the enzyme TPST2, tyrosine protein sulfotransferase 2, that mediates the sulfation. And these are some of the details for what we do. But it's basically what we're doing is we're injecting into the muscle this AAV capsid. The muscle expresses ECD4 that's properly tyrosine sulfated because it has enough of the enzyme. And that goes out of the bloodstream. And then we do sort of escalating IV doses of a sort of a very stringent challenge schedule to test how effectively this can block infection. And here are our two sort of published results. The first one we did, this is a SHIV88. So these are, what we're doing is, we're inoculating an IV, escalating doses of a model virus SHIV that is basically SIV with the HIV envelope glycoprotein. And you can see that all 4 of our controls were infected eventually, and that we can continue to double the dose, and we never were able to get these animals infected. In fact, then they shut down the primate center where these studies were being done, and that's why those ended. So this is also an example to remind you that SIV MAC239 is probably better adapted to as host and therefore harder to protect against, but we did very well against that too as well. We had 8 controls in this case. At 40 picograms of SIV MAC239, we infected all of those 8 controls, and we kept going. And finally, well, not finally, in the next inoculation at twice the dose, we were able to infect one of our controls that had the lowest ECD4 expression, kept going, doubling, and finally got all 4 of those animals infected. So I have to say, though, that this is very good prophylaxis data. This is sort of, I think if the conventional vaccine with this kind of data would be very kind of pleased with this result. More than pleased, they'd be, that would be really exciting. But if we sort of drill down into the SIV MAC239 result, first of all, what now I'm showing you is how many micrograms of protein provided this protection. So at our lowest concentrations, 4 microgram per mil of ECD4, that was when that animal got infected first, of course, after the 8 controls. And then it more or less went down the list, although one of the items wasn't completely consistent with infection. But we use this sort of data as an indication that if we can retain 10 microgram per mil of, or let's say 12 microgram per mil in a human, we should be able to protect them from most HIV challenges. Now, 12 microgram per mil is an interesting number because we have 12 milligram per mil of IgG in our bloodstream. So to pull out a bloodstream, measure your total IgG concentrations, 12 milligram per mil. So that's 1/1000th of your total IgG can protect you from what should be a very robust, in this case, SIV challenge. The other thing interesting about this is we also did we looked at the viral loads. Even though these animals were infected with higher doses, double, triple, quadruple, I guess 8 times in one case, doses of this SIV, the set point and the peak and set point viral loads were a couple of logs lower, indicating to us there was continuous pressure and no escape within the time frame we're talking about from ECD4. So I think that was encouraging. there's a faculty member here named Maurizio Martins, an excellent junior faculty named Maurizio Martins, who's been doing similar studies only with infants. And so this is the same kind of study, except that he's orally challenging the infants. And again, he used conditions in which all 6 of his controls are infected. And interestingly, one of his ECD4 inoculated groups, animals was infected again with, now in this case, about 1 microgram per mil. But you can see, again, very good prophylaxis, 6 and 6. And again, the story repeats. After, animals, that single animal shown down in red on the bottom right, that single animal has markedly lower viral loads, logs lower viral loads than the other 4 control animals, despite having received a higher dose, a higher challenge dose, in this case of Shiv 88. So again, same story. Very nice prophylaxis. When there is breakthrough, there's lower viral loads. So that gave us confidence to start some experiments in which we would actually lift ART from animals that had been infected for a long time and then been put on ART for a long time. And in this case, again, Shiv 88. This is a study by Matt Gardner, originally in my lab, now at Emory. And what he was able to see with relatively low ECD4 expression. So remember, even we're saying prophylaxis requires 12 microgram per mil. And unfortunately, the ECD4 expression levels were lower than that for this study. And part of our work now is to get those expression levels higher. But you can see even with those expression levels being low, we were able to suppress virus. In many cases, so what you're looking at is with these sort of dotted bar blips are in some cases very high blips of virus that also went down in the presence of ECD4, something you don't see in a normal infected animal after you lift ART. And on net, at the end of the experiment, most of the viruses were quite low or below the levels of detectable, with the expectation they would continue to blip along the way were we to continue this experiment. So our goal now, of course, is to see if we can get higher levels of ECD4 and get a more robust suppression after we lift ART in these animals. So that's where we are now. So where we really are now is NIAID has supported development of 5 cell lines based on these new ECD4 variants that I'm telling you about. Presuming that they're safe in non-human primates, these will go into human studies of infected but controlled individuals. We also hope that these new sulfopeptides and some capsid modifications that I haven't told you about will help get our ECD4 levels higher to the point where we can suppress an ongoing infection. And so as I say, our primary goal here is to achieve robust suppression of Shiv and SIV MAC239 after removal of triple therapy with antiviral compounds. Okay, so this is the acknowledgement for that part, which overlaps significantly with the acknowledgement for the second part, which I'm going to jump into very quickly. This is the sort of our SARS work. We've initiated our SARS work. Well, let me start here. Sort of our entree into our SARS work began with our discovery of the receptor for SARS-CoV-1 back in 2003, and I like to say, while that epidemic was ongoing. So there were still people being infected with SARS-1 when we were able to identify the receptor, which was a fun trick. But the truth is we did it through a really stupid method. And what we did is we found cells that bound the part of the S protein that binds the receptor, that mediates entry. And we also used controls 293 cells that did not express whatever it was binding. And we immunoprecipitated. And we identified it with mass spec. And that was it. I mean, it was it was the first time we had, people had done it for viral receptor like that, but it was a really, it's kind of an obvious way to do it now. And a number of receptors have been identified this way. But I like to remind people, mostly say graduate students, that this wasn't really possible too much earlier than that. This was 2003. in 1999, we really had first access to full, complete human genome. So, this would have been hard if we didn't have a human genome to compare our peptide fragmentations. And then it's also important to remember there was some important advances in mass spectrometry, both in the hardware and in the software for identifying proteins using peptide fragmentation and the human genome to identify proteins. So both of those things were necessary to be in place before you could sort of play this very simple, stupid game. But anyway, we pulled it out, we identified it. It turned out to be this protein called ACE2. Now, the other thing we did at sort of the same time is we defined the receptor binding domain for the molecule. And this will be important later. We think it's very important, actually. And this, just to remind you, this was done and nobody had any understanding of the structure of the SARS S protein at that point. So we just, hacked back the S protein in both directions while measuring its ability to bind ACE2, simple as pie. And we zoomed in on this little fragment, about a little less than 200 amino acids. And this little fragment actually behaved very well and bound the receptor better than the full, say, S1 domain. Probably just simply was better behaved in solution. And working with Steve Harrison and Fong Li, Fong Li has had a whole career now in doing beautiful work on coronavirus structures. We were able to solve this structure in complex with the receptor. So you can see this little RBD fragment in complex with the receptor ACE2. And just to introduce ACE2 a little bit, you see that it's a peptidase, and you see that cleft in there. It looks like Elmo or something sometimes to me. And that's where the enzymatic activity is. The enzymatic activity of ACE2 is not necessary for its receptor function. And so just quick intro on ACE2, it's angiotensin converting enzyme 2. As I'm sure you all know by this point, it's necessary for SARS-1 and SARS-2 to replicate. It's a zinc metalloprotease, critical for regulating blood pressure. So a lot of people take inhibitors of ACE1, which blocks, which prevents ACE1 from driving blood pressure up. But the activity of ACE2, the enzymatic activity, is not critical for viral entry. And in fact, it counteracts the vasoconstrictive and blood pressure raising activities of ACE1, is expressed where exactly you would imagine the SARS-1 and SARS-2 receptor would be, meaning your nasal epithelium, your lower respiratory tract, where you get put into the hospital when it does, but also gut. Remember, the coronaviruses are really gastrointestinal viruses at the certainly when they transmit from animal to animal. Also, your heart and your kidney, and it really tracks very cleanly to the pathology that we see. So, for example, we see a lot of kidney pathology. We don't see liver pathology. ACE2 is on the kidney, not on the liver. Interestingly, it's expressed more when lungs are insulted. It's more broadly distributed in smokers. So I always try to get people to stop smoking. Also, it certainly provides some of the explanation for why children are less susceptible to SARS-1 and SARS-2 infection. There's just a lot less of it in the lungs, in the lower respiratory tract, but not in the upper respiratory tract, which means that children can still transmit. They just don't get that sick. There's this horrific possible feedback loop because it's upregulated with type 1 interference. So the virus drives up interference, which drives up ACE2, which drives up the virus, which drives up interferon. As an aside, many very pathogenic viruses, you can sort of identify a similar feedback loop. Just to quickly say that, activated T cells, including those that respond to HIV, upregulate CCR5. So just sort of to note that. And then kind of importantly, ACE2 utilization really well predicts whether an animal species will be susceptible to infection. So we're learning about minks. The new viral variants are infecting mice. The original Wuhan SARS does not. That may be an important issue, etc. So also to remind you that ACE2, as we showed in SARS-1, can work like an antibody. You can make an ACE2 version that works like an antibody. And we did that back in 2003 or 4 or 5 or whatever. And it works very effectively. The problem is, and of course it's very good because you can't escape from it. And in fact, the new variants are binding this thing with higher affinity. But again, back to the PK story, it has very bad half-life in vivo compared to antibodies. So in that FcRn mice I showed you before, it has a half-life of about 3 days, whereas a typical antibody might have a half-life of about 15 days. So we and a lot of people are trying to raise the half-life of this thing because, of course, any individual antibody is going to be knocked off by immune escape, but the receptor actually gets more effective, at least in this first pass of COVID variants that are emerging. The question we were very interested in is ultimately what's going to be a better antigen, the S protein or the receptor binding domain, presumably presented in some way that overcomes the limitations of providing small proteins or small peptides as antigens. And so that's it's an interesting theoretical question because, and you can, you probably have your own opinions. So the first thing to say is, of course, the S protein is going to provide a lot more epitopes than just the receptor binding domain. Okay, that's all right, but many of those epitopes are not neutralizing. And in fact, 90% of the neutralizing epitopes that have been identified or neutralizing sera, and 90% of the activity neutralizing sera has been shown to target the RBD alone. That's a problem and a good thing. It's a problem because that means that the virus has one place it can work very hard to escape from, and that will be an effective escape. Another reason why you might think that the S protein is more effective is because it provides more helper epitopes derived from the virus. So if you just present the 197 amino acid RBD, that may not have enough helper epitopes to really drive the immune memory forward, especially say on a boost or maybe having followed an infection. The other thing that people, and because of the greater number of epitopes, people tend to have the attitude that that the S protein will be more robust to escape. That means that if you put an S protein against the Wuhan virus variant, you'll be more protective against, say, the South African variant. I'm sure most of you are familiar with these terms, but these are as you know, emerging variants that are distinct, tending to escape our current vaccines, and potentially more pathogenic. But anyway, you would think that there would be more conserved epitopes on the trimer than there would be on the RBD, and therefore that's better to limit immune escape. But unfortunately, those conserved epitopes are also the ones that are non-neutralizing. So again, most of the neutralization is with this receptor binding domain, and there's a little bit on the blob that I'm showing you next to the RBD called the NTD, about 10%. That's actually moving faster than the RBD in the new variants. So now the other advantage of the RBD, apart from the fact that, again, I keep saying 90% of all neutralizing activity is targeted there, it's easier to produce as a multimer, especially as I'll show you if you tweak it. And you can make many more RBD targets per gram of protein if you're doing a protein vaccine, or per amount that you're producing from a cell. And again, it's easier to predict and combine to address emerging variants. So as I'll show you, we work with multivalent vaccines that's an easier game to play. So the first thing we did, now it's almost a year old now, is we conjugated the full-length S protein trimer, or we just conjugated the RBD. The first question was we asked, is the RBD any good as an antigen? And the answer is yes, it's very good. If we do it under conditions in this case in rats, where you kind of put your thumb on the scale, make sure you get a good antibody response. You get a kind of a blowout antibody response that's at about 2,000. It's about 1 to 2,000 dilutions, or better than 1 to 2,000 dilutions for those of you from following the COVID vaccine literature. So it's a very good response. Another way of sort of quantifying it is it's analogous to the neutralization you would see if you doped naive sera with 100 microgram per mil of ACE2. So it's in fact a little better than that. So maybe on the order of 200 microgram per mil ACE2. It's two different ways of sort of getting a fix on the potency of the response. So the next question we asked, which I was jumping to, was we wanted to ask which of these is going to raise more potent neutralizing responses. And what we did was we put trimers, S protein trimers, soluble S protein trimers, or just the RBD, and we chemically conjugated to keyhole lymphocyte hemocyanin, standard carrier protein for vaccines, and using a sort of a standard saponin-based adjuvant, much like that which is in the Novavax vaccine right now. We tried to get as close as we could to an apples-to-apples comparison between the S protein and the RBD to raise neutralizing antibodies, and we did so in two formats. One is KLH on one side, and the other is in the context of a 6-dimer on the other side. This is sort of a newer, more modern scaffold for presenting antigen. Again, we chemically conjugated both the RBD and the S protein. And as you can see over here, it turns out that the 6-dimer was better than KLH, and that the RBD was better than the S protein in this context. So, there's a lot of issues about whether it's truly apples to apples, and I'll show you some other experiments that sort of reinforce this point. But at first pass, with our first effort to get an apples to apples comparison, the RBD can surpass the potency of the full-length S protein trimer. Okay, but there's a real problem with what we were doing here. Everything was a conjugate. And the real problem is that you try to make and what we're really trying to do is get to a fusion protein. A fusion protein you can express as a single polyprotein, a single protein construct. So for example, if you want to make an mRNA vaccine or adenovirus vaccine, you can't chemically conjugate that in that context. And the problem is that the wild type RBD is a mess when you try to make it as a fusion protein. And that's basically, to my understanding, why all the vaccine manufacturers essentially gave up an RBD. So that most of them made some sort of RBD fiber to trimer that barely got out of the cell because it didn't really fold well, and it sort of aggregated and did not happily get out of the cell. So what we took upon ourselves to see if we can improve this, retaining the immunogenicity while improving its ability to get out of the cell. And this is just to make the point, I'm sort of jumping ahead here, but to make the point that the wild type RBD now as a fusion with a 6-dimer scaffold can be detected in cell lysates, but it cannot be detected in supernames, in cell supernames. It's either because it's aggregated or not being produced in the cell, as you can see, it's much lower levels within the cell, or because it's aggregated outside of the cell, not getting through our filter. But we've made this thing called GRBD that does so, and I'll tell you about that next. So what is the trick of GRBD? Well, the trick of GRBD is we actually frankly tried a lot of things, and it turned out that if we engineer 4 glycosylations into the RBD, in addition to the 2 glycosylations that are already naturally present, we get a much better behaved protein. And so, I'm just kind of showing you this, and this is the sequence. We've already done our IP game here, so I'm happily showing you the sequence. And I apologize. There's some phones coming through here. So this thing works much better. Now, how do we get to this? How do we sort of down select on this? What we did was we created a system in which we fused the RBD to something that would hold it onto a cell, transmembrane domain. And then we asked the question, how efficiently can it be presented on the surface as a dimer? And the effects of GRBD were less pronounced in this context as they were in later contexts, but there was still a very good down selection. criteria. You can see that both if we measure the amount of expression on the cell surface or the amount of ACE2 binding on the cell surface, indicating the foldedness of the RBD, the modified one does much better. Okay, so we know it expresses better, and that we already, and I'll jump in ahead again, tell you that makes for a better immunogen in the context of a genetic vaccine. What about its intrinsic immunogenicity? Let's say we made the same amount of the wild type RBD and this modified RBD, and we just provided in the context with where the wild type RBD could actually be made from an FC dimer. And the answer is, nonetheless, despite that, the GRBD, and actually this was sort of a happy surprise for us, created a much stronger immune response. We didn't expect this. The reason we did this is we wanted to show there was no loss in activity, but this is a robust result. We have two reasons why this might be the case. First reason is that maybe the glycosylations are limiting the immune reaction to the otherwise hidden part of the RBD, the hydrophobic hidden part of the RBD. Or the other possibility is through aggregation or just incorrect folding, the wild type is just not presenting enough properly folded RBD. But for whatever reason, it's inherently more immunogenic than is the wild type RBD. But of course, our original intention was to get something to be pushed out of a cell in an mRNA or an adenovirus vaccine. To do this quickly, we just weren't up to speed to making high quality RNA, we turned to DNA vaccines. So what we did was we took a range of constructs and we made DNA vaccines of this. But before I show you that to you, I just want you to see that as we go up the ladder of multivalency, meaning the scaffolds that we're using to present the RBD, we get better and better relative expression of our modified RBD, our GRBD, compared to the RBD. And as a consequence, we assumed, and that turns out to be the case, that the higher expressing GRBD construct, when made as a DNA vaccine, would provide greater immunogenicity. So these are just in different contexts. Now, I just want to draw your attention to RBD ferritin. This is H. pylori ferritin. It's a 24mer. It's 10-fold higher expression than GRBD. And what you can see is that, and so I'm going to show some results. And that happened to be, in this context, our winner. Now we're going forward with further scaffolds, but right now that for published or publicly available data, that's our winner. And these are the data for that. So here we're showing direct comparisons between this modified RBD and wild RBD. First on FC dimer, on a fold-on trimer. So keep in mind that fold-on is exactly what people would, the blue line where it says fold-on is exactly what people were using to evaluate the utility of the RBD and compare it to the S protein. And of course, it's not very good, and of course, is better if you make a better RBD. But you can get better and better by increasing the multivalency of the scaffold that you're using to present the RBD. So this 12-mer that we were using did okay. Ferritin did better. The Mi60-mer was kind of a crash and burn because it did not, first of all, it didn't do barely anything with the wild-type RBD, but it didn't express as well as the ferritin, even though the multivalency was higher. So of course, there's a combination of inherent immunogenicity, expression level, and probably the valency as well. And T-cell help and other factors, of course. But the real interesting thing here is, in the same context, we're presenting the antigen that essentially exactly the antigen in a DNA context that Moderna provides and BioNTech provide in an mRNA context. And you can argue that there's a big difference between electroporating DNA versus injecting mRNA, et cetera, but in this context, this S protein provided very poor immunogenicity compared to our RBD-based constructs. And this is another way of sort of presenting the data. What I'm showing you now is just how they did with our wild-type RBD and the ferritin, and again, showing you that this is the IC50 in terms of dilution. and dilution factor, and you can see that the ferritin did much better. And I'm showing you sort of a model of ferritin presenting 24 RBDs in this context. Again, almost the same data again, in which we're just comparing our GRBD contracts. Again, ferritin's the winner, although not by a whole lot, but a significant winner compared to DNA form of the Moderna vaccine. Now, there's a lot of caveats on that. We're going forward to MR vaccine. There's a company, that mRNA company, that's doing these comparisons now directly, and we hope that it looks as good in their hands as in ours. But that's kind of where we are, except that we are right now been testing a series of other constructs, and some of the excitement there as we push forward on the RBDs, and we've been able to lyophilize some of them and reconstitute that without loss of at least either multimerization or ACE2 binding. So we hope they also retain their immunogenicity. It would kind of be a fantasy to be able to provide people a powder they can reconstitute with sterile water. Anyway, the take home here is a multimeric RBD probably is, I think almost certainly is, a better immunogen than those that are people are now using with the current full-length S protein. And that's all I have to say on that topic. These are the people I want to thank on that. All these people kind of mix and match both on HIV and on COVID, but I have to really single out Brian Quinlan as our leader in the COVID field, and the guy who's really been sort of leading the charge both intellectually and in laboratory work on making all these crazy scaffolds and RBD constructs. And that's it. I hope there's a little time to take questions. Well, there is time to take questions. I kind of raced through that. Yeah.
Michael Bertsemler, really terrific presentation. On your modified RBDs, how are you expressing them? Are you expressing them in 293 cells?
So basically XP293 cells. We're moving over to SF9, the sort of baculovirus system,
right?
In an effort basically to mimic Novavax, and we'll see how we do there. Frankly, I think it's the game we're going to win. And then just to be a little bit overly confident, it may be a Betamax versus VHS issue, whereby a superior thing does not necessarily break through into the market. Reflecting some overconfidence on my part, and it's not just our vaccine. A lot of people are kind of, as you might be aware, are pushing these kinds of concepts in various ways. I think I like ours because it's a single protein fusion construct. But the really interesting question is whether a Betamax can beat VHS in this context.
And I wonder if those recombinant proteins could be used as targets for looking for antiviral therapies, as targets for screening.
Oh well, yeah. I mean, in a sense, it's like I don't actually see the advantage over just a plate with a lot of well-folded RBD. But you're absolutely right. But the one thing is the well-folded RBD, which we do better than the sort of conventional RBD, while reserving the key, kind of preserving the key thing. So I should just cut out my vaccine efforts and go into like antibody screening efforts, because we can make a lot more RBD per plasmid than somebody else.
Yeah.
Yeah. But so I see what you're saying there. But I basically think an ELISA plate or something like that, or just some sort of immobilized magnetic bead or something might be more useful than putting, I was also thinking of the scaffolds, but the RBD itself might be useful. The scaffolds may not be.
Yep. All right, thank you.
Thanks, Bert. Are there any other questions? I see a couple things in the chat. I don't know, Paul, if you want to just unmute and ask your question, you can.
Let's see.
Yeah. Yes. So I was just from the first part of your talk, whether the sulfotransferase that creates those sulfotyrazines have been explored as a drug target. Great.
We originally thought back. So I got to say, we saw those sulfotyrazines on CCR5 in 1999, and so it's just one of these long history things. so we talked about those kind of things back then, but and back then we didn't have a sense of the range. You know, sulfotyrosine is actually sort of a constitutive modification of a lot of immune important molecules. And it's also, we showed a little later, an important component of about 1/1000th of your antibodies. It shows up in the CDR3 of about 1/1000th of human antibodies, including some key HIV neutralizing antibodies.
It may be even greater than that because it's very difficult to detect by mass spectrometry. It's isobaric with phosphate, and there are all kinds of problems. So it may be a very prevalent modification.
Excellent point. But the way we sort of looked at that is we looked at an immune repertoire and came up with what we thought was a reasonable definition of the motif plus the other requirements, meaning long and flexible. So we could have made our requirements a bit too stringent. But I will say that it appears to me by inspection of the D chains of antibodies that the immune system seems to have biased itself to propensity to do so. And sulfotyrosine is a great thing because it's highly polarizable and can adapt to sort of a changing target, and yet provide a high amount of binding energy. So yes, actually, when I give sort of the full HIV talk, I do a long spiel on antibody sulfation and how those motifs are very, very common in the D regions of the heavy chain, encoding the heavy chain genes.
Okay, thank you. Thanks.
And we actually have a question from one of our brilliant undergrads. Toki, do you want to unmute and ask your question?
Yes. Yeah, I read an article about heterologous prime boost vaccination strategy.
Yes.
Like we use 2 different vaccines instead of using one same vaccine for 2 vaccination. What do you think about that?
I think it's I think we're about to enter an era when those questions are going to be asked and answered with high intensity. The first 2 ways to think about it. One is forgetting the variation in the virus, forgetting the fact that there's a South African variant and a California variant and everything, just sticking with one. Is it better to say prime with the full S boost with the RBD? Just to ask that question, that we don't know the answer to that yet. Is it better to boost with different scaffolds or different helper epitopes, or the same epitope with different vaccines? And the other question I think is going to be fascinating is, what is the best way to, let's say, just for simplicity, let's focus on the RBD, where we can present multiple different RBDs on these multivalent scaffolds. Is it better to get breadth out of an antibody response by serially starting with, let's say, the Wuhan 2019 variant RBD, and then going to the UK and then the South Africa and then the California? Would that get breadth? Or do you want to do it as a mixture at the same time? Or as we kind of suspect, is it better to even include it on the same multivalent construct? And the reason we think that may make the most sense is you'll be engaging the B cells by, the reason multivalency works, at least in part, is because it cross-links B cells more effectively, and they're sort of primed to see that. Will you be preferentially amplifying the B cells that are responding to both? And so we like that idea, but it's still kind of unproven there. Although I think Pam Bjorkman has some data on mosaic vaccines in which she does a spy catcher, scaffold plus spy catcher with the RBD, making that point to some extent. But for all this hell we've gone through the past year, we're entering this amazing era of human experimentation on vaccines that, if you pay attention, is going to be so illuminating, both for the immune system and for how to make good vaccines and how to think about that. So I think you're asking a great question. I don't think I could ever give you a definitive answer at this point, but I think all of us will know in about a year.
Yeah, because we have HIV, which mutates a lot, and we have lots of variants. And then for like other virus, we have very few variants, and like SARS-CoV-2 is somewhere in between.
Yeah, well, I think so. I have to say that in my worldview, SARS-CoV-2 and those classic coronaviruses, actually all coronaviruses, are at one end of this spectrum where the HIV is on the other end of the spectrum. We're all worried about variants now, but these variants are coming up relatively slowly, considering the number of infections that have been happening. And so the way I understand the world is the following. The coronavirus genome is huge, 30 kb, whereas the HIV genome is on the order of 10 kb, as is viatomol, kind of the flu. And so to avoid catastrophic mutations, the coronavirus has to be more precise per gene than do these other viruses. And in fact, they have well appreciated proofreading functions. So the intrinsic rate of mutation is lower. And as a consequence, the rate of mutations per infection, per replication is going down. But of course, we've got millions of people infected, for weeks and weeks and weeks, multiple replication cycles with this thing. So we're, yes, now beginning to see mutations. But I will remind you that there are more mutations in a single HIV infected person than we have seen in the entire year-long COVID pandemic. If you do the, S protein to HIV-OM comparison. So I wouldn't overestimate the rate of mutation. And in fact, I like to say we're in a period of history for the first time we can get ahead of the mutations because of the rate perhaps of turnaround of mRNA vaccines, maybe protein vaccines, and because the virus is indeed slower. But part of the game, as I remind people who are worried about public or people who are, not wearing their masks, is that we need to really get the number of infections down, because that's the one controllable parameter that will allow us to limit the rate at which new variants emerge. And we're in this terrible box right now. So we have vaccines that are 70% effective, or if you get one dose, and a lot of bad things, a wonderful environment for promoting escape. So I also described this year as the year in which we're moving from where adaptation to host is the dominating driving force in viral COVID evolution, SARS-2 evolution, now to immune escape is the driving force. And I'm afraid we're going to see mutations faster and faster, because those immunopressure will be more directed against those places where the vaccines are having the most impact, meaning primarily, again, the RBD, and secondarily, the N-terminal domain, the NTD. And as far as I can tell, nowhere else.
Mike, do you think you're going to see a difference in the generation of these antibody-driven mutations that we think might happen using the RBD strategy, or a different type of selection?
Yeah, no, I think I think you're right, and that's also super interesting. First of all, take pressure off the NTD, mm-hmm. which means the NTD will start moving slower, which is one of the disadvantages of targeting the NTD. The other being that the antibodies don't appear to be as potent. They don't appear to drive down neutralization to completion. And there are far fewer of them that are, when everybody pulls out the antibodies, they're always, the good ones are always RBD. Mm-hmm. So, and sort of behind all this is the question whether the NTD is playing an independent functional role, mm-hmm. Or whether there's, the reason antibodies neutralize is they're sterically blockading the interaction between the RBD and the receptor, or whether the role is accessory, like promoting association with a sort of interchangeable attachment factor or something, you name it. So there's some really interesting questions happening there. And as you were telling me, we kind of suspect somebody's going to come up with a, as you were describing back for older coronaviruses, kind of suspect people are going to come up with one or two one-off antibodies that target other parts of the S protein, but they are, I think they are less likely to be important from the point of view of trying to raise them with a vaccine, just because, yeah. So I think I very much like the overall strategy of targeting the RBD. It's also intellectually simpler to think about. There's really only about 30 amino acids to worry about. 30 amino acids of the 197 because those are the 30 that are going to vary, that would change in epitope in a way that that'll be on the surface. They can vary, and they don't simultaneously knock off ACE2 binding. So your problem is intellectually more accessible if you want to worry about variance to the RBD than it is to worry about variance to the RBD plus the NTD plus the other things.
Sure.
And I'll say one last thing: NTDs are acquiring a lot of deletions right now.
Yeah.
And those deletions are a very fast way to knock off a lot of antibodies. It's a great question. There's going to be a whole new evolution if people do take up the RBD vaccine. Yeah.
Thank you.
All right, we have time for one more question, and Dr. Brian from St. Louis University crashed our party because I invited him. James, do you want to ask your question?
Sure. So it's a little bit, or it's back to the antibody question for COVID-2, and that's, do you think by presenting the RBD as a multimer that you're increasing the, you're just increasing total antibody, or do you think you're increasing the antibody epitope diversity?
We're, first of all, removing all the distracting epitopes. And you said decreasing diversity. Yes, I think we're decreasing diversity remember what viruses want to do. They, I'm sure you know, but what viruses want to do is they want to put up epitopes that don't harm them, that are that the immune system recognizes. And so we think we're eliminating a number of epitopes that are essentially useless, and then focusing on those that are presumably neutralizing, that are neutralizing. And we might even be doing that in the context of the RBD, where we've sort of coated the backside, the part that interacts with the rest of the S protein, the part that's not available for anything, with glycosylation. So they're not even seeing, they're not even targeting this otherwise potentially very immunogenic region that would not be accessible on the full S protein. So I forgot, there was a second part to the, but anyway, did I answer the question?
Yes.
There was a second, yeah, okay.
Well, Mike, thank you so much for that. That was really tour de force.